everybody, and welcome. Today, we will be introducing a very exciting initiative that we've started recently, Horizon Variant Search. We've opened it global access, and it will be a huge knowledge base for both rare disease and cancer. Today, we have our CEO and founder, Dr. Muhammad Yudin. He will be walking us through the variant search. We also have Maurice Grihan, who is the genome software specialist. And I'm your host today, Mariam Dessouki. I'm the Science Communication and Marketing Officer.
Thanks, Mariam.
Thank you, Mariam. So yeah, it's a very exciting day today because this is the first time we are bringing out a knowledge base for the global clinical genomics community from GenomeArc, and we are super happy to provide this free to use access of our database that we've been working on for years. So without any delay, I would like to take you to the search page and show you how it works, and want to show you how actually it can help accelerate rare disease variant interpretation and also cancer genomics. So this is the page that will be that is now actually accessible globally to anybody who is registered to our account, and it's free to register. You can just come to this page and just registration is very simple. Once you register this page into this page, you just can type out the chromosome and the position of the variant. So it follows the basic kind of like format: the chromosome start position or the location of the variant, the reference allele, and the alternate or the variant that you want to interrogate. And then you have to define whether the zygosity is homozygous or heterozygous. And it's a germline, so this database is actually very comprehensive. It can take input for germline variants or somatic variants with different cancer types, okay? So in this case, we'll look into this variant that is actually a germline variant for rare disease. And you can find the format briefly provided here. And also you can cite this, our database through these two papers that we have published recently. We have validated our algorithm in multiple cohorts. So this is not just the algorithm building efforts. We do also have sequencing cohorts that we employ too in our proprietary databases. So once you type out variants and then you just click the search, but before you do that, you have to log into the system. And when you log in, so you should be able to have full access to the server. And while you click search, it will show you the entire UI that shows whether this particular variant is located in what position in the chromosome browser. In this case, it's in the QRM of 24.3 location, impacting the STN1A gene, which is a known gene for different neurodevelopmental disorders, specifically for epilepsy and autism. So this is a variant that our algorithm predicted is likely pathogenic, and also it shows the ACMG code. And also what are the known disease reported in the literature, including the nature of inheritance for the disease itself, for the known disease. So for this particular variant, it's known to be impacting global developmental delay and epilepsy, as I showed. So you can even, make it bigger just to read out the known disease even with more clarity. And we also shows like if this variant been reported in any somatic or tumor or in any germline pan cancer. So for example, this has been reported in breast cancer and lung cancer. This variant might appear may not have any consequences, but it is also bridging the gap between different genetic conditions. On this side, you see the gene insights where the PLI scores, the constraint index scores, which defines how kind of haploinsufficient this gene is. And also it shows if this gene is part of the carrier phenotype or ACMG secondary phenotype, and the transcript that we are using for this particular variant. Okay. More on that, we have a pharmacogenomic insight for this particular variant, for example. So for rare disease, we know there's not much drugs available. So this particular insight actually helps, clinician or scientist to know what's been out there or been reported at least. So these are some of the inhibitors for SCN1A gene that have been reported. And it also has some RX number if there is available in our database. So these are proprietary database from GenomeArc that we are providing within the free access. On top of that, you see if there is any particular patient in the clean bar. This is a regular clean bar kind of output. So this is listed as pathogenic in some of the patient in clean bar, and these are the phenotypic IDs. And also, what is the variant frequency in different populations? So you can see this is NOMAD plus, because we have also a custom variant frequency from our own research. So these are different kind of ethnicities that shows variant frequency. So this particular variant is pretty much absent. So you get all of this information. On top of that, we have variant rare disease information. So this particular variant actually also been reported in one of the studies that we curated in our database that reported to be de novo. And you can actually go into the study itself where it's been reported. And it also shows you the GWAS, if there is any GWAS, disease association reported for this particular variant. And also we look at if this is insertion, you'll find tandem repeat information on this site. And there are some other features that are exclusively for premium or enterprise version of Horizon. And the interesting thing is, if you look at the gene, this gene you might not know or want to know more in the literature, you can just go down and you can see what are the new kind of literature came out for this particular gene, and you can read it out from our website, and it will show you. So this is a very recent publication, came out like a month ago. So all of this recent literature on the gene also been listed here. So this is how we are providing a very comprehensive overview for rare disease genetics. And if you think about variant of uncertain significance, we will be able to provide more of these, curated samples from our rare disease database, which will be very informative for our user. So any clinicians or graduate students who are using this platform now will be able to tap into our proprietary databases. So there are about 8 proprietary databases that we employ into this unique search server for the global community to access and use. So if you want to look into, for example, the somatic variant, where you can actually type out the variant, determine the zygosity, find out what kind of cancer type you're looking at. So this is kind of, it has a bit of a different UI. So it actually identifies the AMP tiers for you. So for example, we are looking for a variant that is somatic in non-small cell lung cancer. which is impacting the FGFR3 gene, and this particular variant is in VUS tier. And also it shows what are the drugs associated, FDA-approved drugs for this particular gene and variant. And also it shows if this gene variant been associated in the literature on different type of cancer. So yeah, it's also been reported in bladder cancer, lung cancer, and different other type of cancers, because FGFR3 is actually a pan-cancer gene. It's been reported in multiple different cancer types. And then you have the germline information as well, like before, and also the gene insights. And again, the ClinVar and all this other information that I showed you. But one interesting thing is we also have a proprietary database for cancer. And you can see for this particular variant, we have so many new variants being listed, and what kind of cancer type this variant been reported to. It's not just for non-small cell lung cancer, it's been reported to other type of cancer as well. And the relevant PMID also been given, for example. You can read those papers if you want. And you can also access all these variant data from our page. So these are all information that we create within our databases and update every 3 to 4 months. Again, if you want to know what kind of literature is coming out for this particular gene, it shows you the literature down below. See, specifically the latest paper that's been reported on this particular gene. So we also have, a LLM that is actually only can be activated through subscription, the premium version of the software. That will tell you more. You can ask more about the genes and the variants within your workstation, and it will identify, the relevant information that you're querying. We also allow multi-omics integration, so single cell and proteomics integration is being implemented in the premium version. And that is a full-fledged software, not just a variant search page like this. So it's more comprehensive. And we also have a active clinical trial database. So because a lot of this cancer and rare disease patient will require to enroll into clinical trials due to the lack of treatment options. So this clinical trial information will be very important for the clinicians. So this is what the cancer somatic profiling looks like for each of the search variant. I want to show you the last part, so where you can actually, search variant that are structural variants, basically impacting that are very large and impacting many genes. So when you look into this variant, so this is a germline heterozygous variant, doesn't have it's not somatic, and when you search this variant, the first thing you see is then a different number of genes that it is impacting, and these are the rare genes that are known to be reported for a rare disease in this case. So for any large variants, you will see a list of genes that are actually identified already for you to decide if it is, if it is kind of important or not. Then your pathogenicity prediction comes in. But here is the interesting bit. We also have pharmacogenetic information for those genes that have actually drug information, but it's not telling you to use these genes, but rather making a link between those genes and what's been known. We also have horizontal structural variation database where if there is a similar kind of variant exists, it will show up. But in our database, this seems like a unique variant, and it's not. It's very unique variant to maybe for that particular patient. But if it is a common rare disease variant or pathogenic or VUS variant in our database, you see additional sample, which is very powerful for VUS especially. And you also have this structural variation control population frequency that we actually provide. So this control we kind of curated through our research programs and identify what is the frequency of these variants in that control population. This control population is very comprehensive in terms of ethnicity and technology. It incorporates high density microarray to whole genome sequencing variants. So basically what we are seeing from this variant search server, that all of our needs for rare disease and cancer, you can identify different types of insights, and it's free to use. Every month there will be a renewal of your access to the server. So yeah, we are very excited to open it up for the entire world, and we'll go from there, and we will keep updating if there is some glitches or some modification that needs to be made. We'll be continuously doing that every few months. So yeah, so, do you have what do you think, Maurice and Mariam, about the user experience for this search variant server?
From my perspective, it gives just an amazing overview of the variant and the functionality of our GenomeArc Horizon platform. So what it's really doing is giving you a snapshot into the key insights we can uncover with our various underlying databases, some that are open source, such as ClinVar, and then some of our proprietary databases. So I really hope that this supports researchers in their, understanding their variants better.
Yes, I completely agree with Maurice as well. I think it's an amazing initiative that will help a lot of researchers and clinical geneticists. And I do have multiple questions that maybe we can discuss together to show the impact of Horizon. And also, I think me and all other people are curious how it works and how it seamlessly brings these results. So maybe we can go over these questions and sort of go over the platform and see the potential of this, the full potential. So first of all, the first question is, what clinical guidelines does Horizon follow? And what sort of databases does it integrate in terms of proprietary databases and other kinds of databases?
Yeah, so that's a great question. So if you look at the variant search server, so we actually, for rare disease, we give you the ACMG code, and for AMP we give you the AMP tiers, right? So ACMG codes are super important for rare disease because it's been mandated by the American College of Medical Genetics and Genomics to follow that code to determine the pathogenicity of the variant. And for AMP, for somatic variants, Association for Molecular Pathology, these are the guidelines that we follow to help prioritize variants for standard of care or for research. For example, tier 1A variants will have drug information that are FDA approved for standard of care. So we implemented both ACMG and AMP tiers, and we are constantly upgrading and updating the conditions and the databases behind this. To answer the second part of your question, so there are multiple kind of modalities that are involved within this search variant server, and one of the important aspect of the search variant is not just the algorithm, it's the databases that we have. We train the databases to provide us the best insight out there, and these databases are curated by the scientists and geneticists themselves. It's very hand curated, a very high quality database that we use to bring this level of insight.
Very nice. Yes, and I think the databases are showing a lot of results that will be helpful to know what other labs have or what literature has already published in terms of existing variants. So looking at
Absolutely,
this,
yes. Yeah.
yes, so looking at the search
Go ahead.
result, what would you say is most important? Would it be the population frequency or the
Mhm.
computational prediction, the functional prediction scores?
I think for different projects is different kind of database that will show the importance. But for at least for me, what I can tell, for example, this particular databases where we show additional samples that have similar mutations, so that adds a lot of power, for what for for is for rare disease we only can diagnose up to 30 to 40%. It depends on what kind of condition we are looking at.
Yes.
For autism, it's maybe 15 to 20%, but for epilepsy or intellectual disability, it's like 30 to 40%. And for germline cancer, we only know around 10 to 15%. So lot of the time we really don't have an answer. So having put forward this the data that we have from our research cohorts, what or in the literature, this adds great value to first maybe solve a VUS, either to benign or to pathogenic, or refine your pathogenic to either VUS or benign. So it goes in both ways. But for me, additional databases are actually super important. On top of that, we are also showing predictions from our own database, what kind of clinical pathogenicity we have. For example, gene constraint index is a very interesting feature that goes hand in hand with haploinsufficiency. If it is red, it's likely to be more haploinsufficient. So there's lots of those aspects that we brought into the server that you will not find in other databases.
Yes, very nice. Now, zooming in a bit on the pharmacogenomics part, because I know a lot of people are interested in that. Can you tell us a bit more how it's curated?
So pharmacogenetics actually, it's the curation. It's a multi-year effort. It's not just like LLM-based curation because you have to be very careful what kind of relationship you kind of put out. So the curation we did for rare disease is basically all the FDA-approved drugs and also the drugs that are being reported in different type of large cohort-based studies. And those studies, we provide the PMID as well. So people can read, and this is the entire, variant search server is only research use. So only for research use. So you can, learn more about the pharmacogenomics insight from those papers. And also we provide links to the FDA approved, announcement and drug detail. So that's for the rare disease. And for cancer, we actually follow very strictly Association of Molecular Pathology guideline. For tier 1, we only provide variant or sometimes genes that have been reported to have a FDA approved drug for the standard of care. So that information in pharmacogenetic insight you will identify. And these are very interesting because this gene to drug or gene variant to drug information is not readily available in most of the platform. So you can find all of this information here in GenomeArc Horizon variant search platform. Yeah.
Yes, very nice. So it focuses mainly on the actionability, I guess, of the drugs.
Absolutely, because we want to move from identifying variants with the cause for diagnostics to treatment options. So this is why we are putting a huge effort to bring this pharmacogenetic insight even with more in-depth knowledge. So that will be updating every few months.
Yes, very nice. So now let's focus a bit on the interpretation part, because I think that's the main value that everybody will be coming and will try to maximize the results from Horizon variant search. So for VUS variants, which is a common problem that faces a lot of clinical geneticists and researchers, how can our variant search server help change that or help with that?
Yeah, so for VUS, actually, what you need, one of the criteria for even ACMG, that you need to have similar kind of phenotypic cases, especially for rare disease, that have the same variants or similar variants. So when you have that, evidence readily available, it gives you strength into your ACMG criteria to move up the VUS into pathogenicity, right? So we are curating VUS from our own research cohorts. We have many research cohorts that GenomeArc is involved in through collaborators and partnerships. And we bring those insights from these different types of cohorts into the platform to help improve the VUS determination, because this is an uncertain determination at this point that needs to be actually concrete in terms of evidence. So yeah, as much as we can bring into our rare disease database, and populate with many patient information, it will be more impactful for us to resolve this unresolved VUS.
Yes. So looking at, yes, I think so. Collaboration and evidence will surely maximize solving VUSs. Now, looking at the other side, which is pathogenic variants, do you think we've already identified the majority of pathogenic variants in the rare disease and cancer?
Absolutely not, actually. So for rare disease, especially, as I said, for some monogenic disease, you have 80%, 90% variant detected, for example, Fragile X syndrome. But that's just one very rare, extremely rare conditions. But if you look at like broader rare diseases, for example, neurodevelopmental disorders, we don't know. Most of the patients don't have an answer, so we don't know the causal variant. So initiative like this will actually help, finding out more diagnostic solutions. And for cancer, we don't know all the driver mutation. So a lot of those cancers, we don't know which one is the driver mutation. So this kind of, proprietary database that we are bringing in, and also the algorithmic development that we are doing, is aimed to identify those causal driver mutations. And also for germline inherited cancer, we only know 10 to 15%. So we believe that that number is much larger. So bringing those information into this database also will help, to resolve that diagnostic odyssey that we're going for a long time with rare disease and cancer.
Yes. Okay. Another question also about interpretation, which I think even though we have guidelines, and Horizon does an amazing job at combining the cancer and rare disease guidelines, the gold standard ACMG and AMP guidelines, but still the process is sort of subjective. So what happens when the platform has a pathogenic variant in rare disease, but then it appears benign in common cancer databases? How can we sort of bridge this gap?
Yeah, so what happens is we really don't know how variants been interpreted in different conditions and how they're related. For example, a variant that is actually benign in somatic, when it appears somatic, might not be at all benign for rare conditions, because if the variant is germline, it may have roles during the early development of human brain, for example, and that germline variants will be pathogenic. So having side by side Germline, variant interpretation and cancer variant interpretation, it gives you that insight that you should be looking at both, not one or the other, how variants been interpreted and different conditions. Yeah.
So it's different conditions, and we need to not everything that applies in one sort of group of diseases applies necessarily to the other one, right?
Absolutely, yes. So the general interpretation is markedly very different than somatic interpretation. Specifically, ACMG is very different than the AMP conditions. AMP more geared towards standard of care, for cancer patients, where ACMG is more interpreting the variants in terms of diagnostics. But we are trying within this effort within GenomeArc, we are trying to bridge that gap, through the use of pharmacogenomics. We are trying to bring more variant to drug or gene to drug information, even if it is, the knowledge is at the level of research or preclinical. We want to, show this information to the scientists and the clinicians.
It's very nice. Okay, so thank you so much, Dr. Mohammed. Maybe one last question, since you showed us a sneak peek of these premium features
that was
on
going
the
to be
website.
my question.
Yes. So since we're committed to providing it free globally, can you tell us what would be the features that would make it sort of a go-to for researchers or clinical geneticists in genomic medicine? And also, can you describe a bit about these additional features that are locked?
Yeah. So this free version is to help students and clinicians with their projects, and also to help resolve the VUS, that diagnostic odyssey that people are going through, so they can come and look into our databases and see what the variants in question in their projects, what it says in our databases. But the premium version is very different. It allows you to do exome and genome sequencing. You can upload multiple VCF exome and whole genome sequencing as a batch or as a singleton, and it is short read and long read compatible platform. And it's not just the only genomics. It's actually a multimodal platform that also take inputs from single cell data and proteomics. But for genomics, it can take, for example, from Oxford Nanopore or backed by HiFi data, 5 million variants, SNV, indels, and structural variation, including the different types of structural variation that we see in long read data. It can take all of those inputs and can give you an answer within 15 to 20 minutes. What are the pathogenic variants for germline and for tumor? So it takes the VCF files and move on to analyzing the variants one by one, and it's super fast, even for a small server. So yeah, the software is available through SaaS, and also independently can be downloaded, and we can deploy into on-prem servers. So it's very secure to use, and it also have a multiple agent deployed within the enterprise version that allows you to take, say, for example, rare disease complex cases that manifest a lot of different symptoms, and you have no idea what kind of condition this particular case have, and those symptoms can be used to create a gene panel, and you can look into the genome later on with great confidence. So what it does is it kind of correlates the symptoms and the genome. You're making pretty much genome to phenotype correlation. So that's one agentic, but we have multiple agents. We have also a prompt that will identify any variants. You can search variants in that prompt. You can identify treatment options. You can ask any question related to the clinical genomics. We've been trained that way. So all of these are packaged into a very comprehensive, cohesive software that with a very intuitive, user interface. Super easy to use. Anybody can learn in 5 minutes, and just go on from there. So it's as easy as that. So that's why the enterprise version should be accessed for genome and exome sequencing projects.
Very nice. Sounds very promising. And honestly, it's very nice that now we have a free version for researchers to try and use. And we also have the enterprise version whenever they want to make it scalable and with large volumes. Maurice, if you want to add anything?
Yeah, no, I think it looks really, really good. But just knowing the enterprise platform itself, like we have a very comprehensive solution for somatic variant interpretation as well, which gives additional insights into tumor mutation burden, clonal structures, etc., as well as some of the features that Dr. Mohammed mentioned. But additionally, we also have the capacity to integrate custom databases. So, if you have locally curated database or cohort data that you want to integrate into the analysis, we have the ability to do that. So the solution is customizable and really robust for any researchers, analysis, or clinicians and their diagnostic decision making, or to support them in their decision making for diagnostics. So, yeah.
Absolutely. So the enterprise version for cancer is very comprehensive. I just want to add into Maurice's comments that we profile the whole tumor, and not just the variants. Of course, the variant insights are there according to MP guideline, but the tumor also been profiled by computing the tumor mutation burden, the mutation signature, which are very important even for what kind of treatment option that might be relevant for, because say mutation signature brings out the pathway that are involved in different types of cancer, and the tumor mutation burden also allows clinician to decide what kind of treatment option they might need to take based on the accumulation of somatic mutation in the entire tumor. More on that, we also actually create a clonal structure of the entire tumor. If you upload just the somatic VCF file from MUTEC2 or any other somatic callers, we support kind of like agnostically any secondary somatic mutation caller VCF can be uploaded, either the tumor only or both tumor and controls. And it will create the somatic clonal structure. So that allows you to see the driver mutation, how it's clonally kind of constructed. And also you can track relapses for cancer if it happens. So yeah, so all of these are packaged in enterprise version, and all of it is happening in 15 to 20 minutes again. Yeah.
Very nice. So thank you so much, Dr. Mohammed. It was a pleasure having you today and showing us the platform. And thank you, Maurice. I think this is a great initiative and it's very exciting. And we are committed to driving this forward and keeping it free globally. So looking forward to what's coming.